Justin Borevitz. Justin, until very recently, was assistant professor at the University of Chicago and is actually in transition now, moving over to Australian National University in Canberra. He will take up a position in January. So put it to me, he left Chicago when the leaves fell, which is probably an adaptive move. And adaptation is what he's talking about today. Landscape genomics for climate adaptation from model organisms to foundation species. After Justin's talk, he'll be around for an hour or so to chat to whoever is interested in this sort of science, and then he'll move on to some other engagements. So Justin, welcome. We look forward to your talk.
Thanks, Greg. It's great to be here, and thanks for coming out to this special seminar. It's a generic title here, Landscape Genomics for Climate Adaptation, so you can fill in your favorite species. I'll start with Arabidopsis, which has been my favorite species, especially out in the Midwest around the Chicago region. And then I'll finish up with some more recent work in the switchgrass, which is a tall grass perennial foundation species of the prairie ecosystems, which actually also stabilizes the sand dunes here and creates habitat for Arabidopsis. And this is a nice intro slide where I used to try to get out of the urban jungle and get a bit of greenery. It's the Indiana Dunes National Lakeshore Park, and it's a dynamic environment where habitats are shifting right out from under the trees, and Arabidopsis lyrata colonizes the foreground there, and Thaliana the background agricultural sites. And so many of our model organisms and emerging model organisms are here co-evolving with the landscape. And so the work in my lab essentially tries to answer a couple questions. This one, genotype to phenotype, is well popularized in human genetics, genome-wide association mapping. And we can try to look at phenotypes at multiple different levels. I'll focus on flowering time, a very important adaptive trait, and the indirect measures of seed set and fitness, but also show you a little bit about growth and development in real time with some new simple phenotyping approaches. I'll skip sort of the omic EQTLs and metabolomic and methylation work that we've done on trying to dissect these complex traits. When we work with populations from the field, we can try to answer another set of questions, some of which is also try to focus on the genes important in adaptation, but populations can be where they are for neutral processes, and then genome-wide markers can help us figure out how populations got there, neutral processes of gene flow, demography, and selfing kind of weedy colonizing species, they can tell you indirectly about the species that move them around. So we'll take a look first at the distribution of genetic variation in Arabidopsis, and then come back to the phenotype. And hopefully we can try to integrate this genotype-phenotype environment story. Especially in plants, the environment has a big effect on the ultimate phenotype. And so trying to study the genotype-phenotype problem really needs to consider multiple environments. In plants' plasticity, the ultimate phenotype is always a combination of the environment and the genotype. And ultimately, together, we like to think about how the assemblage of organisms in that environment ultimately shapes the growing conditions. And that's kind of the first slide that I showed you about the dynamic system in the Indiana Dunes. So local adaptation then is when certain genotypes are going to be filtered as they have preferred phenotypes in certain environments. And this has been a question studied for 7 decades now at least, from Clausen, Keck, and Heisey across the way, and they had this wonderful transects up along the altitudinal gradient toward Tahoe. And when you do reciprocal transplants of several different species they were working on, they often found that the home populations did better in the home common garden. So this reciprocal transplant experiments are very important to establish the genetic basis of local adaptation, but they couldn't answer what's the molecular genetic basis. The DNA hadn't even been crystallized at the time, and how many steps does it take to become locally adapted, and are those new mutations or are they new combinations of standing variation. So those types of questions now we can try to get at. And we've been using Arabidopsis thaliana, which here is growing in these patches that we always thought were clonal patches. They establish disturbed fields. One seed can give rise to 10,000 seeds in the next generation and quickly take over a field. And then we wanted to look at the distribution then of genetic variation on the landscape, phenotyping in simulated natural environments, which is very important to try to understand the fitness consequences of the traits we measure. And we really moved genome-wide association mapping forward in plants. In 2010, when a series of papers came out showing how this works. Then I'll finish up the talk with the switchgrass story, where we're using genotyping by sequencing and looking at genetic diversity in this foundation series. So here we go, genotype on the landscape. And here's Arabidopsis in its local environments. This is a cornfield. Some of the later flowering lines that you can collect when the corn is already established. Other plants are colonizing empty parking lots here and competing against weedy lettuce varieties, and railroad tracks are another common habitat, which Arabidopsis does well in these disturbed environments and eventually gets pushed out by grasses after several years in fallow fields. We also collect in the off-season by scooping up frozen patches of soil and bringing them back into the greenhouse. So this variation through space and time in the populations that we are looking at. And together with Joy Bergelsen and Magnus Nordberg and others, Eric Hollov collected thousands of lines in England, and collaborators in France. We looked at this, basically the sum total collection of all Arabidopsis lines at a panel of 139 markers. It was a quick way, this was before NextGen came out, but we could look at some common markers just to try to identify clones and look at some more recent family level population structures. So we went through this set, and pairwise comparisons then revealed that most lines, especially in Europe, were about 50% diverged from each other. These were common markers, and so that's what we expected by chance under these null models. And there were a stack of clones, so especially in North America, this one variety, which we affectionately call the Heartland strain, and a few clones in Europe. So this kind of took that dominant paradigm of Arabidopsis spreading as a clonal weed and said most of the diversity that we find is at least deeper than we could be picking up with these standard set of markers. Although in North America, when fields are recently founded by multiple individuals, we had another sort of population of natural recombinant inbred lines or natural near-isogenic lines, and these are a fantastic mapping resource that I spent several years in grad school to try to generate manually. Now we know which fields we can go and just collect these lines. So this is the diversity at the global level. If we try to map that back onto where these plants were collected, you can see the red spots are where there's no diversity, essentially the clonal fields. There's a few clonal fields in the US, but by all means they're not all clonal, and there's some that are yellow or white here, which have almost all the lines were completely different from each other. And then another within field sort of common was multiple founders or two genotypes, so that the third plant sort of had a 50% probability of being either of the two that were already collected. And so orange and yellow are quite common in Europe, and there's a few more clonal sites in the recently colonized North American continent. And then the ancestral regions of Western Asia and sort of the Iberian Peninsula tended to have slightly higher diversity. But that's kind of the global view then. If we want to scale down to the local view, here's this red dominant type, this redneck or heartland accession. It was basically seen as the most common type in most fields, although just like in Chicago where there's these segregated neighborhoods where the majority genotype is the minority genotype in some populations, there's also quite a bit of segregation in Arabidopsis thaliana at the local level, and some potential interplay between different types that migrate in or that are ousted as conditions change. So here's some of those clonal fields. Here's a field with two different types. Here's a field that are developing these recombinant inbred types where two genotypes are being mixed, and some very high diversity sites that they look like they could be in Europe as well. So North America then isn't a bad place to study certainly colonization genetics and population processes in Arabidopsis, which nobody kind of believed at the time. And even the temporal variation, there's quite a bit to do here, but this red type is only dominant in the early May '05, and the next year March, but by June it was kind of common again. So there's seasonal dynamics and even long-term sort of dynamics. This site Joy Bergelson had collected in '93, and I went back in 2005, and you can see that that red type is sort of coming in, but the purple type is still hanging around. So this just gives you a bit of feeling for the temporal and spatial scales of genomic variation in Arabidopsis, and it does map onto the ecology of the location. These are phenotypes, days to flowering, under constant conditions in the growth chamber, and preferentially late flowering lines tended to be found from more shaded or woodland environments where they would overwinter, and early flowering or rapid cycling lines were preferentially found in open fields or disturbed kind of sunnier environments. And so at this stage I should introduce that there's really two common life history strategies: this overwintering approach where seeds germinate in the fall and then flower in the spring, and they often require a series of cold treatments to get them to flower, versus the rapid cycling or more weedy type that does well in the cultivated fields. And the tractor would probably come in between this stage and impose strong selection for rapid cycling. And so there's many derivative changes that can convert late flowering into rapid cycling, and those are kind of coexisting life history strategies. So let's focus now on the phenotypes, and we're going to do that in environments that are important, and we're going to think about what is the right set of genotypes to start looking at phenotypic variation. And so that actually is a very important problem. Which genotypes you look at is going to affect the genetic architecture, and so we wanted to think about high-power mapping populations, and I'll show you the empirical relationship of all those 5,000 lines in a second, but this is sort of a summary where there's local kind of patchiness and extreme population structure and some deeper branches. And so there's two strategies then that can be taken to develop high-power mapping populations. One is to just focus on certain regions, or what's being called now regional mapping or reg map lines, and so just go back then maybe and fill in some more collections from these relatively less structured samples where you've got reduced genetic and allelic heterogeneity. Of course, you're not going to answer questions about the standing variation at the deeper level, and to get that we have to take a more synthetic approach, which is to sort of prune out a lot of these short branches and come up with sort of a more star phylogeny, if you will, of distantly related individuals that might have diverged from a common ancestor much earlier and then have recently colonized the whole European range. So to get at the early variation, you can create these synthetic global hat map with balanced population structure. And then to break down that remaining population structure, this is nested association mapping approaches, or intercrossing some of these lines. So that's the strategy then for high-power mapping populations. And empirically, this is what we did. This is the big Heartland accession, which we collected over 1,000 times in the Midwest. That would really confound our association mapping, so we dropped that as well as some of the other clones that were identified 50 to 100 times. And 1,800 unique accessions is all that exists in the Arabidopsis database, or in our community collections. And we wanted to break down some of the family structure in here, so we took a higher level pruning and came up with this core set where most of the individuals are relatively equidistantly related to each other. So this we call the global hat map set, and lots of people have ordered the lines. We took, at the time, a custom SNP tiling array was the tool of choice. It's 250,000 common SNPs here from a resequencing study, a Perlogen array-based resequencing study. So we knew these were going to be good markers, and now we're doing this with next-gen sequencing. And you don't need to sequence the whole genome because of linkage disequilibrium. You can impute a lot of the missing lines. It's pretty straightforward to multiplex multiple lines in a single. But we're at 1,300 of those 1,800 ecotypes now have been typed on this array. So we have a deep global hat map set. And we wanted to do the phenotyping then in not standard laboratory conditions of fixed temperatures and light and long days or short days, but in geographically defined seasonal climates, which I'll show you in a second. We wanted to have conditions that sort of span the native range from the south Taça del Mar, which is an interesting population where major alleles for salt tolerance are segregating and show a nice climb. And a northern population in Sweden, although there are some farther up and more recently derived there, that southern Sweden might have been even a refugee, and there's a lot of diversity in here. So these two conditions then define day length variation, temperature variation, and even light intensity variation throughout the day and throughout the year. So we have these sometimes affectionately called time machines, or seasonal growth chambers, climate chambers, where we can dial in perfect sunrises and sunsets exactly as would be expected on an ideal year. Any field trial may have different results because of the particularities of one year, but set climate where the daytime cycling and the days getting longer much more dramatically in Sweden than in Spain, and day-night high-low temperatures, the evening night temperatures, that important vernalization signal. So these are the predictable seasonal hues that Arabidopsis would feel, and we grow these lines in here and do our phenotyping. And custom bulbs actually cost more than the $200 cameras that we set in there to try to record in real time growth and development. And I'm just showing you from one environment here, 6 different, 3 replicates on this side and 3 different, so these are 3 replicates of 2 genotypes. So you can try to estimate by eye the heritability. And we count green pixels every 15 minutes as we go through here and use a little background filter to call those. There's the trait of rosette area, which is shown in white, circular area, and the difference between those, which is kind of compactness. And so they're pretty well behaved across the replicates. This now is real plant behavior, if you will, with their motion and time lapse. And it's the other genotype that has longer petioles and slightly different shape. So at every 15-minute interval, we can calculate the 3 traits and then map QTLs through time. And I'll just show you here the heritability that it's very low at the beginning stages, and then which line is bigger and which is smaller sort of changes through development. And so the heritabilities get stronger as the plants, and then when they grow out of the pots, the trait kind of drops down as well. So this is genotype by environment and genotype by development. And so we ran QTLs then. This is the first of many genome-wide association plots, which I'll show you. 1, 2, 3, 4, 5 chromosomes, and this is, each spot is a SNP on the 200. Well, in this case, it was 160,000 common variants off of the microarray, and every point then is the minus log p-value of dissociation with leaf area. And just if you fold this out of the board and color code these significant lines in blue, then we can look at the genetic architecture. There's 3 major loci controlling variation and growth at the early stages, and 4 major loci, maybe 5, controlling growth later stages. So we can really see that the genetic architecture of size depends on time, and I could show you another plot for Sweden. It also depends on environment. So there's a very intricate way of dissecting these complex traits. So let's focus then on flowering time. This is the days till flowering in 4 environments. An early planting, a spring planting is in March or in May in Sweden. And this is a summer planting in April or in June. And so June planting in Sweden, most of the accessions flower right off the bat at about 25, 30 days. And then there's a long tail of lines that we're expecting to have a winter treatment, and they never flowered in our study. But this same global HapMap diversity set grown in early spring conditions, there's really an absence of early flowering. In 50, 60, 70 days, by 80, 90 days, they're all finished. So within a month, this global diversity set is homogenized to have a single unimodal distribution. And in the intermediate environments, we see more or less 2 groups, but they're really starting to emerge in this condition. So this is the 2 life history strategies and 1 life history strategy. The trait is quite plastic. And if we just look at the GWAS here, there's a locus FT that's important for spring flowering time, but not a lot else. And that's because the phenotypic distribution is really homogenized by the environment. But in Sweden summer, where there's a big separation in that genetic diversity is revealed, the common known alleles at Frigida are important. Those are coming up. And another important set of traits on the end of chromosome 5. So some of these are known genes, and there turned out to be 12 major loci that can explain 50% of the phenotypic variation. So this is a large component of the missing heritability is really identified in these balanced global HapMap samples. And I've sort of ordered the 12 QTL then by their environmental sensitivities and color-coded them by the map colors to a certain extent. And so these QTL here on the left, they are very sensitive to the time and less sensitive to location. So maybe 8 leaves or 8 days of flowering time variation in the spring conditions, but in the summer conditions they have almost twice the effect. So these are sensitive to when in the year, when in the spring they germinated, whereas SFT3 really doesn't have any sensitivity to when, and some of these others have less sensitivity. They're more sensitive to where. So the difference between Sweden and Spain was very important. And so you can imagine that different accessions then have different allelic combinations at these 12 major QTL, which allow them to flower at the appropriate time across the range. I'll show you a couple more data slides that suggest that. One is that flowering time and seed yield is got this interesting correlation. Most standard growth chamber conditions show a positive correlation that sort of goes on. But in our conditions, we see this split correlation where there's an optimal flowering time and seed yield relationship. And it's earlier in the summer plantings. But if you flower too late, sometimes there's a little spindly stem that comes out of the side, and it gets cooked in the warm Spain conditions. So seed yield and flowering time are important. The other thing is that we have a nice correlation with where the plants were collected. So in blue here is a p-value distribution for latitude by genotype, and so that's a long tail with very few loci showing a relationship with latitude, but 4 of those were the QTLs that we identified. So 4 out of 12 flowering time loci show this nice climb across Europe, and the Columbia allele delays flowering and is more preferentially found in northern Europe. So relationship to fitness and a nice climb on the landscape are suggesting that these are adaptive. All right, so this last couple slides then I want to show you about fall plantings. And so we cranked the chambers down to about 5 degrees and gave them a couple months of cold and shorter days, and tried September and October planting. And you can see that the growing season, as defined by days above 6 degrees, gets shorter in the future conditions. And so here we simulated, we did a global warming experiment in a single location and just changed the temperature. So one chamber ran 2040, the other chamber ran kind of contemporary conditions, 2010, and the lower shelves are about 1 degree warmer, so we filled in these intermediate thermal blocks. And so we've put our global hat map set of lines into these environments, measured flowering time, and lo and behold, October contemporary planting conditions, nice tight distribution. So again, this global diversity set is showing very little phenotypic diversity. It's hidden by the environment here with between middle April and May, the whole set flowers. But what happens in the warmer conditions? Might we see the other life history strategy coming out? Well, so far it looks like Arabidopsis is not seriously impacted by climate change. It's a couple lines maybe flowered in the winter and would be sort of selected against, but for the most part, warmer conditions, a little earlier spring growing days, and the means shifts a little bit. Maybe there's some rearrangement in here, some rank order variation. No, it's pretty much a replicate. In fact, when we saw this, we thought, boy, not much to report here. Maybe we don't have to worry about this thermal regulation. On the other hand, in September, it was a completely different story. So earlier fall planting really did the opposite. This is even more extreme than that Sweden summer planting, where half of the lines were rapid cycling right out of the box, and the other half waited and overwintered. And very few were in the wrong location. So this is a tremendous amount of plasticity as well as environmental sort of splitting of the phenotypes. And so in fall, in contrast to in summer, what you get is a decision that has to be reached whether we can get reproduction in before the end of the year or whether we're going to wait out and go for the overwintering strategy. So this is quite a bit of diversity that's been seen. In the future conditions is also similar, that we see these two groups. A little bit of a shift. There's a few more plants in the rapid cycling group. Now, what happens if you look at the pairwise comparisons? There's a big shift. Some of the lines that were overwintering with 2 degrees of warming completely reverse in a qualitative way to rapid cycling. It's about 10% of the lines are coming out of one life history strategy and into another. And so we can do genome-wide association mapping on the main effect of flowering time as well as on this thermal sensitivity. And we find 5 new seasonal flowering loci, including 4 that were previously known, and 4 new thermal sensitive flowering time loci. G by E point. So here's a couple of the major effect QTLs that have a big phenotype here, 25, 30 days difference in flowering time. These are linked to the Frigida gene as well as some other known candidate genes. And here's just an example of 2 of the 4 G by E or thermal sensitive SNPs, where it really has no phenotype in the contemporary conditions, but out in the warmer chamber and warmer shelves, and likely in the future conditions, future climates, this SNP would have a big effect. So this could be an example of selection on present-day standing variation in future environments. And here's the opposite example, where an allele delays flowering in contemporary conditions, but now promotes flowering in the future conditions. And there were good candidate genes for both of these. The internal hormone flowering signaling gibberellic acid and the heat shock protein pathways both came up here. So if we just think about plugging these SNPs in, how much do they explain? This is one allele now. If we color code that pairs plots that I showed you before by genotype at that one SNP on chromosome 5, most of these lines that are doing the reversing have the non-Columbia allele here. And so you can see in future conditions, if you have the Columbia allele, they really aren't so sensitive to temperature, but you are quite sensitive if you have that non-Columbia allele. So together then, can we try to predict what the phenotypes would be in future conditions? And so if we train a model based on 3 of the 4 blocks, leave out the 2055 block, and use the genotypes and the phenotypes to predict what the phenotype would be in the future, that fits pretty well with what we observed. And so population structure explains about half the variation, and these 4 G by E plus 4 a priori and 5 thermal flowering time QTL can really recapitulate the model. And this side we can see the prediction of genotypes that were left out of the model. So if we use all 4 environments but 90% of the data to specify the genotype, phenotype, environment model, then we can predict with pretty good accuracy the missing genotype. So if you grew a line that you'd never seen before, but you knew its genotype, with 95% accuracy you could predict the flowering and in future environments. So that's the story for Arabidopsis then. We can look at patterns of variation across the landscape to understand about recent demography and population turnover, new migrants, recombinants, natural populations of mapping lines from a few founders. Complex traits, either flowering time or using time-lapse cameras to really get at real-time behavioral variation. And letting the trait then tell you about what are the developmental stages. And in the end, we see QTL that are sensitive both to season, location, and temperature that are common across the species range and likely important for adaptation through new combinations of standing variation. Now this approach doesn't get at adaptation through new mutations, but what we do have in that Heartland background, we do see phenotypic variation on a clonal background. So there is the ability to use the same genetic test now with next generation sequencing to go back and look for phenotypes that are due to brand new mutations, since we've accounted for common variation here. And so at this stage, everybody often says, boy, you can do a lot with the Arabidopsis. It was the first plant sequence, thousands of labs working on all over the world. You have all the tools at your disposal. What about other species? And what about more complex environments and perennials and long-lived plants? And so we started out looking at switchgrass, which is a popular bioenergy crop now, but it's really originally was a forage crop and is a foundation species in the tallgrass prairie. Ranges from Canada to Mexico and from the Rockies all the way to the East Coast. And so has tremendous range. And is that due to plasticity or is that due to genetics of adaptation? There's been a little bit of low density genetic data to suggest that there is sort of clines east-west with rainfall and north-south with day length. And we wanted to look in this local environment and compare that to the continental range. So we did that using next generation sequencing, and it was just a couple runs. We multiplexed 36 samples. There's really no good reference genome in switchgrass. It's an allo tetraploid that has octoploid derivatives up in the northern range. There's 2 known subspecies that roughly in the lowland group and in the upland group here that have been isolated for a long time and really could be called separate species, although there's been some suggestion of hybrid zones. And then we had another 20 plants coming from our local populations here in the Indiana Dunes for comparison. And so for $40 or sample, or even less now with the newer sequencing capacity, we can get at tens of thousands of nuclear loci and the whole chloroplast genome. And so briefly, this is genotyping by sequencing that's also been done in Ed Buckler lab. It's slightly different from the RAD-tag sequencing approach. It's easier. We're using a single enzyme. PST was a good one for us to reduce the complexity. And then we adapt, so like digested genomic DNA and then ligate on the Selecta sequencing adapters. And we introduced custom barcodes in here. And then PCR amplify in pools the fraction that's amplifiable, which is like 200 to 600 base pairs. So you get a little bit of junk comes out, but mostly this amplifiable range. So that gives you the reduced representation so that we can look at multiple reads on multiple individuals in these stacks. So bioinformatically, We can generate a pseudo-reference. We used CLC Bio to generate that. It's a simple assembly because you're only looking at about 5 to 10% of the genome and a bunch of reads from a bunch of individuals. So the color of the barcode here represents which individual, and then the SNPs can be identified. This would maybe be homozygous for one allele. This red individual is heterozygous, and this other 3 individuals have the ancestral allele. And so we're both doing de novo genotype calling as well as so de novo SNP discovery as well as genotype calling, and we can find a lot of common variants as well as quite a few rare variants that are in 1 or 2 individuals. And the last thing to mention here is that because this is a real kind of population sample, we look and filter on SNPs that are true polymorphisms because they have alternative genotypes across samples. And a lot of times, especially in polyploids, you get kind of a heterozygous genotype in all individuals because there's 2 copies and one of them has one variant and one has the other. It's not really a genotype, but we use the large number of individuals to confirm that it is segregating. And so you can get rare variants that are heterozygous in just 1 or a couple individuals. And then buried in here, because the ligation is not perfect. In fact, the first time we did this, we just gave the digested DNA to the core and they did blunt end ligation. And so we had a lot of read-through, quite a bit actually. Here's 600x, 500x coverage of the chloroplast. And there had only been studied 4 genes for chloroplast variation, and there was about 10 known SNPs and a couple indels in switchgrass. But what we came up with was 100 or more, 120 or so SNPs and a half a dozen indels across the whole genome, which gives us quite a bit of power to look at within population level diversity. And so chloroplast is non-recombining, and we can develop a pretty good phylogeny. So here's that upland-lowland split that had been known even from the limited chloroplast sequencing. And that's variation that predates the Pleistocene really and goes back farther than the 2 subspecies of indica and japonica rice. We also identified this new deep split within the upland group. We have labeled here A and B, and this turns out to be quite old. And then there's lots of sub-chloroplast haplotypes that also come out in this clade that could be used to discern more fine-scale variation. And notice that there's a few plants from the dunes in both of these A and B deep groups here, which is pretty interesting. That's the phylogeny is confirmed with the indels. And if we think about trying to date this nucleotide divergence based on the maize-sorghum split, then there's quite a bit of error bars on this time scale, but it sort of roughly corresponds to the interglacial cycles, that these sort of predate some of the big ice shifts that would have resorted the prairie grasses into different refugia in Cuba and northern Mexico, and then recolonization of the landscape, including the dunes site here in Michigan, which has happened many times since the glacial tides come through. Now at the nuclear level, what we did was kind of a simple trick that Jonathan Pritchard suggests. Select randomly one read as the allele at each locus, because it's tetraploids in here, and so we had to deal with the genotypes are a little harder to call, but if we just permute the population structure analysis using one read per individual, then we can see how robust these are. And obviously the upland-lowland group comes out pretty strongly. These are 3 lowland individuals. And the upland group, including lines from the dunes, and here's one from Michigan, show this sort of graduated population structure so these are more eastern types, including the dunes, and then the more western types. And so at the nuclear genome, this upland split is now being homogenized, right? And so these may have diverged long ago, but at the nuclear locus, they're starting to come back together, which is excellent for mapping variation. If their whole genomes are diverged, then you really can't get at the causative basis. But if there's deep ancestral diversity that is now being reshuffled in situ, and this is a very local populations now, the nuclear genomes are relatively homogeneous, even though the chloroplast genomes have diverged. And plants that are basically right next to each other have chloroplast genomes half a million years ago. So that's for the genetic diversity of switchgrass, and we wanted to try to look a little bit at the phenotypic diversity, and I'll just kind of conclude with this, that the biofuels or bioenergy plots, or what we really think the value is in the below ground carbon sequestration. There's big phenotypic differences in these cultivars and in these accessions. And so the right variety for yield in the cellulosic kind of model is often when you move the lowland varieties, which have longer growing season and more extended rainfall. When you move up north, they grow vegetatively and eventually get killed by the frost, but you could harvest right before then. And so biomass, above ground biomass yield, and we do have carbon data below ground coming for this, differs by quite a bit in these cultivars. And so you can go with the USDA recommended genotypes, but we really don't have any idea what the best variety is for any new environment. And so we wanted to think about mixtures of genotypes in much the same way that David Tillman has this prairie hay biofuel, which is mixtures of different species. We were thinking of mixtures of different ecotypes and accessions for switchgrass. And so this is a large now from Google Earth. You can see the size of these plots. They're like a quarter hectare in size each. And so they have different diversity treatments and 2 nitrogen treatments. And so the way we did this large common garden was to block it in 3 units. There's a cultivar, so this is like monocultures, and then a eco pool of multiple types. There's a local type and the 2 kind of elite varieties that were recommended in upland and lowland. And then with big blue stem, which is another species, or with Canada wild rye, which is a C3, so mixing warm and cool season, and the prairie mix. So can we look at genetic diversity and species level diversity effects on productivity in these plots? And so we're finishing up the 3rd year now, and the take-home message is the prairie plots were all right. Sort of air bars a bit overlap with some of the others. Fertilizer helps a lot. That's not a big surprise, but you know, it needed to be shown. The local type, which is what a lot of conservation groups would recommend, was the worst. And maybe that's because they're not recommending it for biofuel, but that's what the local variety does compared to the 2 elite lines. And the pool, which has this lousy one as well as the others, did as well as any of the others, and maybe, you know, was even higher here on the unfertilized treatment. So in a sense, then, if you have genetic diversity, then the best type will sort of be in there by chance and do well. So there's really no kind of detriment to yield by having extra diversity, and having extra species diversity didn't improve things a great deal. So the next thing I want to show you is from this corner right down in here, where we did smaller plots but a lot more genetic diversity to really kind of get at diversity within big bluestem and switchgrass, and evaluate the levels of genetic diversity as having more diversity compared to monotypes improve things. And so this is a mixture of the 2 species with more genetic diversity, or the monocultures of either big bluestem or switchgrass and extra genetic diversity. And the results then are here. So Blackwell and Cave and Rock were those, Canlow were those recommended ones, and there's quite a bit of variation between plots. So the noise then is another factor, and so from the individual plots, then you can predict what the average biomass should be in the mixture, and the observed biomass tended to be greater. That's what the increasing slope is for both switchgrass and the switchgrass bluestem pools. So we're seeing not extreme, but you know, certainly a significant improvement both in the reduction of variation and in the better than average yield of genetic diversity in these biofuel kind of experiments, large-scale reconstruction experiments. And this is a scaling, so it's still significant if you scale the data by the best performer, and so the mixtures are yielding slightly better than you'd expect in the best performer. So that's the story for today. You heard about the Arabidopsis, local adaptation, high throughput phenotyping, and a little bit of a landscape level view, both at the genetic and phenotypic level. And that's the wrap for the Chicago story. I'm on my way now with Craig down to Australian National University, which is the capital here, and so we got a couple postdoc slots that if you're interested, let me know. I got to give acknowledgments to the Arabidopsis flowering time work to Yan Li, and she's in Nanjing now, starting her new lab in China. Xu Zhang did a lot of genomics work that I didn't talk about with tiling microarrays, and also she did the camera high throughput phenotyping work. And Alex Platt, along with Joy Bergelsen and Magnus Nordberg, were instrumental in the early diversity studies where we came up with these core hat map stuff. Jeff Morris has now moved on. He decided not to come to Australia, but he went to University of South Carolina, where he's going to continue working on switchgrass, and Paul Grabowski is going to finish up in Jonathan Pritchard's lab. They've got plenty more populations and switchgrass genetic diversity to look at, in collaboration with Mike Kassler from the USDA Wisconsin, who's kind of the master of switchgrass diversity, and Mike Miller, who's out with Argonne at the Fermi Labs, the terrestrial ecology group there. So I'll leave you with this little time-lapse video done by postdoc Tim Brown, who is coming to Australia, and this is his high-throughput phenology from the dunes landscape. He's got a camera sitting out there now for the past couple years, and it takes pictures 3 times a day, 100 frames in each, and they get stitched into one big gigapan. He calls this gigavision, where you can zoom down now into the actual plant and figure out exactly when it flowered. And so we can sort of use this ecosystem level phenology data to be a little smarter about how we're collecting plants. Here you'll see some cocoons come out in the spring, 1, 2, 3, and by June then they're gone again. So you really need to, we can't be in all places at all times, but the cameras can be there for us with just unbelievable resolution. You can almost see Arabidopsis librata plants in here. And this is all up on the website, so you can pan and zoom and play. There was some wormwood growing in there. Here's a cocoon. Another little Daliana plant. All right, so thanks for your attention. I'll take any questions.
Thank you. Turn on the lights, please. Someone's close to the lights. And there's plenty of time for questions. Maybe I could start while people are thinking. So in that population mixture experiment, was that the lowland, what you call the lowland versus upland clades, or was that different populations that are already naturally admixed in
So this
the northern clade of the
this level? This has, yeah, there's one lowland here, and then the rest of them are upland. So just randomly, some of the 2 and 4 and 6 plots would have lowland and upland.
That's what I brought to mind is in a lot of plant restoration work, particularly things dealing with the rapid environmental change or,
right.
or revegetation of mine sites and so on. There's tremendous concern about outbreeding depression, possibly over-concern. So
I've heard that as well, and I'm not sure. I certainly don't think our data, if anything, our data would suggest that increasing the genetic diversity would give you a better chance for restoration and yield success. Okay. I'm more worried about inbreeding depression from habitat fragmentation, and especially there's very few little prairie postage stamps that are left intact, and that's where a lot of these cultivars then come from. And the cultivars aren't so bad. They collect a big set of seed at one site, and they plant it out and expand it, and then that's the cultivar. So it hasn't gone through like single seed descent or something, which we proposed to really evaluate genetic variation. And the DOE said, why would you ever want inbred lines? You know, as a Arabidopsis geneticist, I can't imagine not wanting inbred lines. But I worry more about inbreeding depression than outbreeding depression.
Somehow that's a message that needs to get out to the practitioners.
I'm glad that.
Other questions or comments?
Yeah.
What other kinds of phenotypes did you look at in your Spain versus Sweden simulation beyond the growth rate and the flowering time? Was it just this camera setup, or did you have other phenotypes?
And so the short answer is we looked at lots of phenotypes, but not very well yet. And so the first thing that we could analyze from these stacks of image data is green pixels. And so we just kind of went with that to push it out there. But I could play you another video that shows that the petals, the time of day that the petals open and close is also captured in there. And it's so neat. It's just sort of, we never really looked at that carefully before, but if you go in the afternoon, the petals are closed, and in the morning they open up. And this leaf rhythm trait and the petiole elongation is really dependent on the environment. So Spain versus Sweden, when the petioles elongate. The short answer is that there's so much plasticity that each shelf gave us a different result. So when you do that GWAS plot that showed the 3 genes in juvenile stage and 4 loci at the adult stage, on the next shelf it was another genetic architecture. But those are the same shelves that differ by 1 degree Celsius, which is important for flowering time. So, you know, good chambers, lots of phenotypes, and I think core set of lines is really another important tool. That if you had random populations, maybe half of them would be clones and they wouldn't really give you a good balance. And the other thing is that if you have this global diversity set, there might be too much going on. There's a lot of major alleles, but they sort of dilute each other because you've got the species-wide set in there. So we wanted to go wide at first, but now groups are starting to focus on either the Czech group or the Spanish group or the French group or the Swedish group, you know, and the genetic architecture is not as complex. But it's almost like every environment gives you another answer, every population gives you another answer, and that's how it works out there, I think.
This isn't really my field of study, but have you considered looking at RNA expression, maybe taking daily samples while you're, you know, doing the photography and just seeing how much gene expression changes on a day-to-day basis?
Yes, and we have, and I just didn't talk about that today. I was going to see if I can find take me a minute to find the movie, but there's some of the movies you can see that we did take off a leaf. We've not done it every day, although with RNA-seq now and, you know, multiplex approaches, you could certainly think about that. We did juvenile and adult leaves and flowers from different environments, from these same plants that we were photographing. So the idea was to try to be able to have the seasonal populations, seasonal phenotypes, ultimately have flowering time with growth and development. And so you have a gene expression profile. You'd like it to be predicting important signaling pathways and even candidate genes that are important in flowering and loci-adapted, you know. So yes, you can do gene expression, and we find a lot of differences. And they're in genes that are not unrelated, they're in pathways. And some of those pathways, you know, with gene ontology, you can find what you're looking for. So the method is certainly interesting, and so expression QTLs we have done. And others are doing that too, but I think where do you go next with that? Is kind of an important question, so no shortage of tons of data. So I didn't even show that today, but we have a couple papers on it you can look at.
Okay, we're coming up to 1 o'clock. We'll get out to 1, so some of you have other things to get on with. Do so, but others who want to stick around and chat to Justin, we have a chunk of time with him now. Please take advantage. Thank you, Justin.
All right.
Think we see that in our lifetime. That's why I love genetics. Here they are doing
You okay with that?
it is amazing. It's the black box is no longer
Right, the black box is put up, and that's for sure.
So what brought you to Redwood, speaking of black boxes?
There's the forest or the most.
She's in there.
That is so
Hey.
nice.
Yeah, that's
So, I own Martin and I have long
Hey,
cabins. And I did a lot of research on this big tree state park.
doing the functional test in the end.
I wasn't in the field of this.
Big sugar pines.
Yeah,
Sugar pines are wonderful. Actually, there's been a lot of sightings about sugar pines just lately. So whether there's actually a larger sugar pine found up
Yes,
in
and that's corrected out. Yeah, yeah.
The
No,
largest ones are always
the Emma Fish and Mixed Model Analysis has population structure.
Oh, I love
That's
it.
the record.
Or taller.
So
not
that
bigger. The biggest one
hurts us a little bit, but not too.
Just come by us and
Okay, all right, take care. Hey, good to see you again. Doing well out here? All right.
0.7 meters of
Excellent.
ability to measure that.
70 meters
Enough.
tall.
Enough to know.
Big trees, and there's one up north that's 7.7. No, they can measure
My
them
wife
Yeah,
was
that's
with
true.
that accuracy these days.
Yeah.
But
if it
they'd
smells
have to
like
measure it
weeds,
next year again because they're all growing really
They
fast.
have cameras.
Maybe smelling, you know, depends on
So
which.
once,
The amazing
I think we
thing
forgot it on the
is
table.
We'll probably pick
redwoods,
some
I've
other,
learned is that,
you know,
there's absolutely no sign
We
of
don't have wild
senescence in
Arabidopsis
the
population.
big eucalyptus is a nest.
we have enough
They
diversity
brought out
for
them
more
inside.
across.
It's taller than
And your students are staying in Chicago?
most
Paul and Tracy are staying.
the organisms they're putting on, adding
Yeah,
this carbon
yeah.
per
I'm a little worried
year
for
of the
Tracy. 'Cause she's
tallest
kind
and
of been on
biggest
her own for a while,
redwoods.
but
They're growing
see how
more
she
rapidly.
does.
than
Paul's gonna go. She's gonna work with Jocelyn.
And they're putting on
Yeah,
incredible
I think they'll
volume
figure
per
something
year.
out. And Paul's
So
got
has
enough
Steve
data.
Siler
He's gonna
gotten you
work
up
with
into this?
Jocelyn.
Oh,
Yes,
that's good.
it hasn't
Brown
got me
yet?
up yet.
Yeah.
The tallest I've ever been
That's
is
good,
I
good. Well,
when I was
I'm glad you came.
when I was still
Well, I'm
maybe 20.
talk with
I
Craig
climbed
about, you know,
almost
the
to the
next
top
projects
of
that we're
a
gonna
citrus
do.
spruce because they're like a ladder. You can just walk and get in them. There's
Can I
just
come
so
over to
many
your office at 2?
branches.
Is that gonna
So I
work?
walked. I got to the top of the citrus spruce studying
Yeah, come. I'd like to see your setup. If you have any, you know,
tree bolts.
comments for cameras and stuff.
That's
There-
right.
And there are.
Yeah, yeah.
And the other thing, I don't know if you know, but a species of salamander
Maybe I
didn't
can meet you
really
back here.
realize.
How
All right.
about that?
Yeah,
You want to come back here in 2 now?
That's